here. Screen. Is that being shared? PowerPoint? Zoom land, do you see chapter 6? All right, good. All right, and then somebody's going to tell me when to stop, right? How far did we get?
Barely. I would stop. I think we're aisle 1 on.
Oh, okay, the individuals.
Yeah.
Oh, right there.
Yeah.
So we have that list, and then we're here, right? Okay. Oh, and it goofed up again. I can find it now. How about that? All right. I think we're recording. I think I'm sharing. I think we're in. Oh, I had it marked in my book. Page 80, right? Yeah, ready for your heme test? When's it? Today? Today and tomorrow.
Yeah, whenever.
So y'all take it tomorrow.
Yeah.
End of the day?
Well, I
Yeah.
thought I was ready and then we had to read it.
11:56.
And you learn. That's never a good feeling when you learn something in the review, is it? New stuff. All right. Hopefully it wasn't like new material. Oh, I forgot. We didn't lecture on this, but here it is. All right, so here we go. Cytokines of the innate immune response, IL-1. So interleukin-1 acts as an endogenous pyrogen. So pyrogen, what does that mean? Pyrogen induces fever, okay? Pyro, fever, heat your body up. So IL-1 being produced gets the body to do its temp. And as your mom might have told you, no temp is good. We want to knock that down, and we want to make sure we treat you with Motrin and Tylenol and keep it down. Your body doesn't see it that way. Your body sees fever as a good thing. Your body induces its fever to respond to growth of organisms. So we know if we increase the temp of the body, maybe it factors into the growth of the organism and slows it. So IL-1 is that inducer of fever. How it does is acute phase response through the hypothalamus. So IL-1 is produced, it goes to the hypothalamus, influences it. That's your thermostat of your body, and it changes. Hopefully A&P taught you that. I hope, hope you got that out of A&P when you went to neuro. It also induces the production of some other things. It induces vascular cell adhesion molecules, chemokines. That's the attractants that bring things to areas. And then it actually induces IL-6. And again, we talked about IL-6 being the key here to COVID, which would be, highlighted. You start seeing that. It includes a 1 alpha, a 1 beta, and also includes a 1 RA. Of course, we got an abbreviation there. Does anybody know what RA stands for? Can you find that? Is it in the little paragraph here?
Is it receptor antagonist?
Yeah, receptor antagonist. All right.
Antagonist.
Yes, good, very good. So they all have something. I mean, alpha and beta are pro-inflammatory, meaning they're going to make your body get into inflammatory. Cytokines produced by monocytes, macrophages, and dendritic cells. Okay, so our phagocytic cells, these three, okay, in the blood, out of the blood, and then around in the areas out away from everything for the portals of entry to dendritics. The ILB, or the IL-1 beta, responsible for the systemic activity attributed to IL-1. So the beta unit of the IL-1 is the one that gives us all the systemic activity that we associate with IL-1, fever, activation of phagocytes, and production of acute phase proteins. Okay, and our acute phase proteins, we'll see those as, C-reactive protein always comes to mind because that's one you may or may not be getting done when you go to the ER. when you come in with fever of an unknown origin and they don't know if you got an infection, they don't know why you're running fever, so they'll do a C-reactive on it. You guys have just learned, from what I hear from him, you have just learned an older way of measuring C-reactive protein, acute phase protein reactions. You remember what that was? Can you remember the test y'all worked on? I know you did it in lab. I heard it. I've heard them over there talking about it. Got a new machine over there that actually helps you do it.
The last.
Sed rates, right. Sed rates is that inflammatory, yay, we know they're somewhere, right? It increases, your sed rate increases and the red cells drop farther if you do a column the old way, right? So that is an acute phase protein where we can measure the activity with sed rate, but we really don't have one to measure. And C-reactive is one they like to measure now. So they can actually measure C-reactive protein levels, and that'll give an acute phase reactant level. So you know how doctors are. Some of them will still order that sed rate because they're so used to doing that. Or they may go to the C-reactive protein so they can get a number and see that rise as to infection or inflammatory response. So we see that. IL-1 definitely plays a role there. The reception antagonist is produced by the monocytes and macrophage. It acts as an antagonist to IL-1 by blocking IL-1 receptor. Okay, so IL-1 receptors are on these cells. And this IL-1, so it controls itself, right? So this RA portion, this RA1 of IL, actually blocks IL-1 from continuing its activity. So antagonist means we stop it, we control it. There's a check to the IL-1. So here's the next one, is tumor necrosis factor. Okay, another cytokine of the innate immune response. Okay, so we've spent a day or two on the innate. Tumor necrosis factor alpha, the most prominent of the tumor necrosis factor family, exists in membrane bound or soluble form. So one of it's either going to be floating or it's going to be attached. It causes vasodilation. So from anatomy, you should have got that is, the blood vessels expand, increasing blood flow. And that's what if you increase blood flow, you're increasing redness, you're increasing hotness, right, to the touch. You're also increasing cells getting there. Okay, so vasopermeability is what we're looking for. Tumor necrosis factor brings that about. It's triggered by the presence of this lipopolysaccharide found on gram-negative bacteria. So everybody but Preston was in micro lab yesterday. You were dealing with a gram-negative bacteria, right? Our Neisseria are gram-negative bacteria, right? Okay. Yay. So you've had that, right? So if I had a Neisseria gonorrhea infection, I would expect tumor necrosis factor to be increasing. If I had a meningitis, I would expect tumor necrosis factor based on the lipopolysaccharide found in gram-negative bacteria. So, and we got a bunch of gram-negatives coming next week. So we had that big micro lecture to kick off our coliforms. It's secreted by, again, activated monocytes and macrophages. All right, what activates a macrophage and monocyte? Anybody remember? We just had a few slides back. IL-1, right? Remember? Didn't it say IL-1 activates monocyte, macrophage, and dendritic cells? I think so. It also can activate T cells through its ability to induce expression of major histocompatibility type 2 molecules. You've already had that, right? So which T cell are we associating with type 2? We'll review. Oh, we love reviews, don't we? Remember, we had a T helper and a T cytotoxic, and we associated them with MHC 1s and 2s. Remember that? Maybe. Anybody? Maybe for a bonus question? Bonus? Might be a good bonus question. You don't want bonus questions, right? Like one bonus question.
Cytotoxic?
Cytotoxic.
Yeah.
Already knew the answer, just throw it out there for me. Vascular adhesion molecules, chemokines, and in a similar manner to IL-1, that's the activation of the T cells, is tumor necrosis factor alpha. Here's our IL-6, right? We said, hey, this one probably playing a bigger role. I think I watched a video last night about T cell immunity versus antibody immunity to COVID. Anybody picking up on that story? It's kind of been hinted at for a few weeks. Is that maybe our T cell activity is ready to roll when it comes to COVID? Not having to wait on the antibody production. Who knows, right? So T cells may play a bigger role in why you're not getting sick. That immunity is attacking the virus quicker than an antibody-led response. You don't know that now. You don't know about T cells. You are all about your T cytotoxics, right? T helpers. But IL-6, again, key to the interleukins. It's going to be a big one playing forward for probably another 6 months or so. Not hearing a whole lot about it because we're not getting a lot of info from the hospitalized patient. Again, it's disappointing to us teachers that like info. We like to hear what's going on inside the hospitals, and we kind of got to get in there to find out because they're not letting that info flow. Because you just don't know. We're not hearing any details as to A, B, C, D leads to death, or A and B leads to death, or A, B without C would come back home. You know, we're not hearing a lot of that. We're not hearing a lot of details of the, as we would say, the mortality, morbidity of the patient that's suffering from COVID. So it's lacking, but maybe it'll be in research papers in the future. And definitely something that we can revisit next semester in lab when we get to virology, when we can start saying, hey, wouldn't it be cool to look for papers that are talking about what actually happened to the hospitalized patient? IL-6 is a single protein produced by the lymphoid and non-lymphoid cells. Y'all just came out of hematology again. Who's lymphoid and who's non-lymphoid? IL-6 is going to be produced by both, right? Myeloid versus lymphoid. Did y'all not pick that up? Is that test not coming tomorrow? Myeloid, lymphoid?
Which is why we're worried,
Okay.
honestly.
This is what I would say. I would say lymphoid, right, line of, development, and this would be myeloid line of development, if you want to think of it as when y'all split off on your little flow chart for hematology maturation, all right? And then there's certain cells we end up with in our diff that came down those lines. Now, at least probably need to know that. Is this your first test? Okay. IL-6 is part of the cytokine cascade released in response to that lipopolysaccharide. And where did we see lipopolysaccharides again? Gram-negative bacteria, right? Okay. It's pleiotropic, meaning it plays many roles and plays a role in the acute phase reactants. And it's triggered by our beginning one, which we said earlier, which is IL-1. So we're going to see these play in a minute. I think we got some pictures from the book that show us how all these interact with each other and with other cells. So we'll put some pictures to the words, but right now we're just kind of working through a multiple choice question about IL-6, correct? Stimulates B cell proliferation and differentiates into the plasma cell, which spits out the antibody. So that is key too. IL-6 is going to be our B cell stimulator. So whenever it gets produced, right, it's going to influence the B cell. And we've already seen that. We've seen that with maturation. We said, hey, here's IL-6. We'll talk about IL-6 later when we get to it individually when we talk about the cytokines. But hopefully you're seeing why this unit's going to play off each other. We got the antibodies, right? And then we're going to talk about cytokines, and then we're going to cap it off with our greatest chapter ever, right? Complement of our 30 complement proteins. It also induces the CD helper cell to produce greater quantities of both pro and anti-inflammatory cytokines. So why would we need pro-inflammatory cytokines? And why would we need anti-inflammatory cytokines? Anybody know? It's kind of a balancing act. Your body's got to attack, but it can't overwhelm. So that cytokine storm, right, has got to be controlled in some way. We can't have like, oh, and then let the body try to figure out how it's going to recover from that. Okay, so it's got to have some on and off switches, and IL-6 is going to influence the CD4. So if IL-6 is increasing, maybe we see the CD4 going into pro-inflammatory, and then when IL-6 starts to back down, maybe we see the CD4 go into anti-inflammatory. So they're talking to each other, they're communicating, influencing different reactions to the cells. All right, so that's IL-1, tumor necrosis factor alpha, and IL-6. Now we're going to talk about a group called chemokines. Again, the innate immune system, they enhance motility and promote migration of many types of white blood cells toward the source of the chemokine. So chemotaxis. So all of these are chemical attractants. So this is the, you want to say this is the cologne or the perfume, or think of it that way. So if these are kind of things will follow that gradient. They will see that chemokine path and they will go back to the source. So it's an attractant to move to an area that the white cell is needed in. Okay, they're classified into 4 families, and they're based on their N-terminal cysteine residue, and we'll just know them as alpha beta C, CX3C, and I mean, if we get this breakdown of chemokine, I think that's the reason we group them all together. But there is a separation of them. Just know that chemokines is more than one. Chemokines are involved in the initiation, and the development of inflammatory response in numerous diseases. So that should be what? That's a common theme of all of these cytokines, right? We're seeing they're involved in the initiation of inflammatory responses. They combine with the cell adhesion molecule and they facilitate extravasation of leukocytes into the tissue, areas of inflammation. So again, another one of those vaso, help the cell get out into the area where it's needed. And they do with the cell adhesion molecules. So what you'll see in the circulatory system, if you hadn't seen this before, is the cells circulating down. There's going to be these molecules that kind of start attaching to the endothelial cells of the wall lining of the vascular system. So that white cell comes in and kind of attaches it, kind of catches it. And then that allows it to slow down in the flow, and then it allows it to get through where the cells, the membrane. So this kind of opens up a little bit, and that cell, as long as it's attached and can get there, then the white cell moves out. It goes out of the vascular system into the tissue. So that's how that works. So when you have, all of a sudden you introduce something to your skin, like under your skin, think of a splinter. Just think of a simple splinter, right? What happens? All of a sudden that splinter's there for a little bit, and it's going to start to swell. It starts to get red, it starts to get painful, right? Because there's putting pressure on the nerves in that area. But what's happening is your immune response is coming after that splinter. And then the first thing you know, you got these pus start to build up around it. That's your white blood cells dying out for you. They're trying to kill parts of that splinter. They're trying to degrade it. So think of that. I mean, you think of something in your skin, and then how does all that happen, right? We know we got some macrophages out, but how would it increase the neutrophils? How would they get out? How would they do that? So
Is it the same way for like a pimple or something like that?
For what?
For like a pimple.
Sure.
The same thing.
Yeah. Yeah. Kind of the same thing. That's why we
Curious,
take.
I know it's a weird question. But
No. I mean, well, splinter, pimple, whichever way you want to look at it, right? A cyst, a bite, staph infection, ant bite. We've all had that, the redness show up, right? All right, so that's our chemokines. Not a whole lot there. We get a whole list of them on 82. Okay, and you can see that we got CC chemokines and CXC chemokines, and then those little molecules we're talking about as for cell adhesions are known as integrins, and that's we have that highlighted in that section. So this is TGF beta, and that is not tumor growth factor, right? So don't think of it that way. It is what? Transforming growth factor. So we hear tumor necrosis factor, you know, with TNF alpha, and then all of a sudden we put a TGF, and some students always will keep that T as tumor. It's actually transforming growth factor B, beta. Has 3 isoforms, 1, 2, and 3. Induces anti-proliferative activity in many cell types. Induces anti-proliferative. So it was originally your book says this nicely, it was originally characterized as a factor that induced growth arrest in tumor cells. Okay, so that's transforming growth factor, because that kind of sounds like what? Kind of sounds like that's opposite of what a transforming growth factor when your mind would be. We would think what? It should be helping things grow, right? Transforming growth factor. Any growth factor should help us all get bigger. This one actually anti-proliferate influences of the cell types. So it regulates cell growth, the differentiation, apoptosis, migration, and inflammatory response. It helps down-regulate the inflammatory response when no longer needed. So this is one of those control factors that we see to the immune response. I think I got another. Yes, this is what I was looking for. Inhibits activation of macrophages and growth of different somatic cell types. Functions as anti-inflammatory for the mature T cells. So this is putting the brakes on the inflammatory response. Okay, inactivated B cells, right? Well, we don't need you anymore. We're going to slow you down. Inhibits the proliferation of the B cell into the plasma cell. If we want to go that far. And it may function as an autocrine regulator to limit the expression of activated cells. So the big take home for transforming growth factor beta is it is opposite of what we've been doing. It's not IL-1. It's not tumor necrosis factor alpha. It is not chemokines. It is, well, maybe, but it's doing what? It's controlling. It's bringing back things. It's stopping the proliferation. It's stopping the activity of all of our innate immune response. Okay, y'all doing pretty good. I mean, this is if you hadn't read the chapter, I mean, it's right here. I mean, we're just step by step through this unit. So what we got next is the last one on our list, which is interferon alpha and interferon beta. And as soon as you hear interferon, as soon as you hear it, virus, okay? Everything else has been, bacterial, but this one is only for viruses, okay? So when we hear about interferons, we're thinking viral, stop the viruses. So definitely a player in what we got going on today. So it can be used, our body produces it, but it can be also used as treatment. You could treat viruses with interferon, like HIV, right? Take interferon, slows down viral replication, keeps that from happening, and maybe we can reduce the virus down to nothing, right? A lot of these things, like I know there was a big push last year for, do you have hepatitis C? Okay, y'all remember that? Like it sounded like the whole world had hepatitis C, you just hadn't been tested for it, so you better run out and get a test for hepatitis C. But one of the things was they say, we got this new treatment, we got this new drug, if you hadn't been treated with anything else yet, okay? So what they were looking for were patients that were newly diagnosed, hadn't had any onboarding of any other antiviral medication, and then take the one we have and watch it work, right? It's really, really effective as long as you hadn't had what? Another treatment. So what's tricky about viruses? Viruses mutate, okay? So you throw something at them, they'll figure out a way to change, okay? So that's what they're meaning. Like before you got treated with that antiviral, this one would have worked. But since you got treated with the other antiviral, now the virus is mutated and ours won't work. That's kind of how the story goes with antiviruses. All right, so the interferon is produced by the dendritic cells. We know that is a phagocytic cell that hangs out. It induced production of proteins and pathways that interfere with viral replication and cell division. And it's so good at this, it activates our natural killer cell. That's that other lymphocyte that we always forget about. We're all about T's and B's, but we always forget about our natural killer lymphocyte. It's activated by the interferon. So who plays a big role in killing virus cells, virally infected cells? The natural killer lymphocyte does. It enhances the expression of MHC class 1 proteins and are active against certain malignancies and other inflammatory processes. So that's nice. It's nice to know we have some kind of malignancy growing in us, or before we even know we have it growing in us, maybe interferon are helping us get rid of it. Mainly secreted by T cells, especially the T helper cell. Okay, whoops, no, where were we? That's kind of weird. We're switching over to cytokines of the adaptive immune system. Sorry, made the switch, sorry. It's not divided up really well. All right, so that's the end of the innate. The innate cytokines. Now we're getting into the cytokines of the adaptive immune system. So I guess the one thing I don't want you to miss is what? There's a lot of interaction between these two. They're both going on. It's not like you got to go, oh, the innate's over here doing its thing, and if we're needed, we'll ramp up. There's cross activity going on. You've seen the names playing in both, right? So here are the cytokines of the adaptive immune system. These are mainly secreted by the T cells, especially the T helper. After they're secreted, we have affect T and B cell function, more directly cytokines involved in the innate immune system. And then we have 3 subclasses of T helper, which is T1, T2, and Treg. So that's our 3 that we have with this T helper story. With the cytokine production. So we're going to be talking about what T helper 1 produces. We're going to talk about what T helper 2 produces, and we're going to talk about what Treg produces. So if you're looking at your text, you will see on page 83, and I think, yeah, here's what I'm referring to. We'll just go over here and start this off. So here's our naive T helper, which is going to move into a different maturation depending on what it captures, okay, what antigen it captures. How's it, this is our antigen presenting cell, and ooh, that didn't really transfer very well, did it? So we've got bacteria and viruses over here with red. I see fungi with 3, and extracellular large antigens like parasites in 2. Okay, sorry that this has happened. I think it's just the way the slide is. Y'all have it better, I think, in your book. So the color coordination is that we have T helper 1s, we have a T helper 17. Well, that's kind of new there. He kind of threw that in on us, Mr. Rector. T reg and a T helper 2. And what you're seeing is over here, you can't read, I can get the numbers here. That looks like IL-12, IL-6, that we've talked about already. We've got, looks like interferon gamma maybe up there, tumor necrosis factor. Got big. Okay. I don't know what I did. I think I got too close to the board. All right. So now we'll go back. Oops. Yeah, there we go. So now when I say there's a T cell receptor on the CD4, right, makes sense that there's a receptor on the T helper cell. Captures the antigen, and then clonal expansion occurs. So the T helper's got to, once it knows which antigen it's dealing with, then it goes into a differentiation to form a different T helper. Okay, make sense? And these lineages are influenced by the spectrum of cytokines expressed in the initial response. So the interleukins that we're talking about influence the T cell, the T helper cell, to move into the different areas. Okay, so this T helper 1, I'll break this out. That's interferon gamma and tumor necrosis factor beta. This is IL-17 and IL-22. This is tumor necrosis. This is transforming growth factor beta, and that maybe hopefully makes sense, right? So let's bring that. I know it's not really seen right here, but that's transforming growth factor beta. And who is the cell that we see with the T helper? What? T reg, right? Makes sense, right? The regulatory T helper cell that brings everything back down is being influenced by the cytokine we just talked about, which is the transforming growth factor beta, who downregulates proliferation, downregulates all this B cell maturation, right? So it makes sense that we associate, this is what I want you to do, associate transforming growth factor beta with T reg. Got to make that connection, because that makes it easy to think about. Right? Because we just talked about down-regulating, and then we got a T regulatory helper cell there that matured because of the influence of the cytokine. And then it's going to be influenced even more to slow down the other T helpers to stop them. So let's look at these individually. T helper 1, interferon gamma. This is kind of how it does its thing, okay? So remember, what was T helper 1 doing? T helper 1 was in our scheme, it was the red, right? It was bacterial and viruses antigens. So bacterial and virus antigens. So if you hear viruses, we do what? We associate interferon, right? Okay? So affects the RNA expression level of more than 200 genes. Stimulates antigen presentation by the class 1 and class 2 major histocompatibility molecules. Activates macrophages to stimulate phagocytic and cytotoxic abilities. And it regulates the action of the T helper cell. And it regulates the action of the CD8 cytotoxic cell. And it regulates natural killer lymphocytes. Okay? All right, so T helper 1 cytokines, interferon gamma. So interferon gamma is not only playing a role in the innate, it's got a role in the adaptive. Okay? Right? IL-2, known as T cell growth factor, drives the growth and differentiation of both T and B cells. So hopefully, this is where and you don't have to go to the other class and tell them you got a better immunology than we did last year. This is the connections. We're connecting step by step this year. Okay. Last year was like, whoa, we didn't even We left the cytokines, we just moved right on by them, okay? Hopefully you're seeing that reason we stepped it the way we stepped it this year is so you know the cells that we're growing up here. There is no reason for you not to know what's being controlled there. There's no reason to not know, that's a better way, what this is, the lytic activity of the natural killer, okay? So IL-2, interleukin 2, known as T cell growth factor, drives more T cells and B cells. It induces lytic activity of the natural killer. It can activate proliferation of the T helper 2, okay? Which we hadn't seen T helper 2 yet. We've seen it, but we hadn't put it in our list. It helps generate IgG1s and IgE producing cells. So, driving what? Antibody production. That's IL-2, interleukin 2, under the influence, the T helper 1, influence in T helper 1 to drive the IgG1 and IgE production from the B cell to the plasma cell. Here's the T helper 2 cell, which is under the activate proliferation of IL-1, right? IL-2 produces IL-4. So if we go back and look, IL-2 is way over here on the other side of our first little page 83 figure we looked at. We see that it is humoral immunity and anti-parasitic. So this is our extracellular pathogen parasites are influenced by the T helper 2, and we associate IL-4 with the T helper 2. So parasitic infections, allergies, autoimmune diseases, okay, IL-4 influence, the T helper 2 was influenced by IL-4 production because the antigen that we found was a big parasite or receptor expressed on lymphocytes and various non-hemopoietic cell types. But the big thing, you're like, what's the take home? The take home is I need you to start putting these cells with their cytokines, okay? Especially in the adaptive immune response. IL-10, because we can pull a board question today and I can tell you I can pick out an IL-10 question, I can pick out an IL-4, and basically they'll ask you which cells are under the influence. So T helper 2, again, way over here on the other side with large extracellular antigens and parasites. IL-10, so we had IL-4, right? IL-10 has anti-inflammatory suppressive effects of the T helper 1. It's produced by monocytes, macrophages, cytotoxic T helpers, T helper 2s. Inhibits antigen presentation by macrophages in dendritic cell. It serves as an antagonist to the interferon gamma to downregulate immune response. So, sounds like who, right? Sounds like transforming growth factor beta. And it also sounds like our Treg, right? So IL-10 is the interleukin that slows down the T helper 4. I mean, it's T helper 2, but it slows down the T helper. Okay, so IL-4 is driving response, and IL-10 is bringing it back, slowing it down. The Treg is next to our T helper 2 in the proliferation. We've already talked about what it's associated with, definitely the transforming growth factor beta and the IL-10. So you just saw IL-10's description, and this should be no surprise that it's right here with us with Tregs. Induces IL-10 and transforming growth factor beta expression on the Treg1 cells in the peripheral circulation. So produce transforming growth factor beta to suppress other T cells. And it also helps establish the peripheral tolerance to various self-antigens, allergens, tumor antigens, transplant antigens, and infectious agents. Right? Because we don't, if our immune system just keeps roaring, we've got consequences to that, okay? So, let's put you in the seat. You are now the endocrinologist, the rheumatologist, and you're concerned that your patient has autoimmune tendencies. Can you see where some of these tests might be worth knowing, some of these levels? Maybe patient has what? Has some antibodies against some of this. And so we can definitely check for antibodies. So a lot of our test menu, especially from the rheumatologist, or I can't remember who had the longest test list. But they're looking, right? They have to screen the patient for everywhere from this levels, right? We could look at levels of each one of these, to actually antibodies that may be against some of these cells, right? You got a Treg cell that's being attacked by antibody, what happens? You lose the ability to slow down the immune response. What if it's a T helper 2 cell that has influence with some IL production? Hopefully you're seeing that. T cell suppression occurs through IL-10, inhibition of pro-inflammatory cytokines. Okay, so IL-10 inhibits pro-inflammatory cytokines. And then there's also inhibition of costomolarity molecule expression of the antigen-presenting cell, which means we're going to keep the antigen-presenting cell from doing its thing. Transforming growth factor beta downregulates antigen-presenting cell function. We're not going to proliferate and more cytokines from the CD helper cell. We're going to tell the CD helper cell to stop. And the immune response is downregulated, and thus chronic inflammation is prevented. That is where we'll go later in this semester, is we're going to head into the autoimmune stuff. And we're going to get into it, and you're going to learn about autoimmunes, because that's where you're going to use immunology the most, because your patient's going to come in needing TSH, just simple TSH level, okay? The reason we're checking TSH level is because they have an autoimmunity. We've got fatigue. How many patients have I seen on the diagnosing for any lab test? It's always fatigue, okay? We're running these things, so we can help. We know we're going to learn more about autoimmunity than you probably ever thought, because we're going to go through them all. We got time. And we already know what's going on with everything, right? We're going to know the background and the principles behind these. And we're going to be excellent autoimmunists, right? Autoimmune immunologist. All right, there's this one we hadn't introduced. He was on the list, or she's on the list, every way you want to remember these. It's the T helper 17. T helper 17, look, it crosses both paths. It's the innate and the adaptive immune up here on our big title. It has a role in host defense against bacterial and fungal infections on the mucosal surfaces. So if we go all the way back to our little picture, this is the blue or purple, whichever way you want to call that. This is the fungi you see, and you'll see T helper 17 basically picking up that purple color for fungus infection or fungus outbreak, whatever way you want to call it, what's going on. But superficial mucosal surface infection. T helper 17 secretes IL-17. Yay, we got a link. We got the numbers the same, right? You're probably thinking, well, that's about time that we have a T helper 17 that secretes IL-17. Can't miss that. If y'all miss that question when it comes up, be disappointed. So you associate 17 with IL-17, and it promotes continuous recruitment of neutrophils, promotes release of antimicrobial peptides, and when dysregulated, have been implicated in pathogenesis of multiple inflammatory disease and severe autoimmune conditions. So not good there, right? We were doing so good. We were doing so good till we read about our autoimmune severe conditions of our inflammatory response. All right. So this should look familiar. Maybe. Heme test tomorrow. These are our hematopoietic growth factors. IL-3, erythropoietin. I wonder what cell is influenced by the erythropoietin. Do y'all know? Which cell would we get more of if we gave our patient erythropoietin? We gave him EPO. The erythrocytes, right? Yeah. All right. How about y'all heard it? Is it Neupogen? Is it Neupogen? That's to increase your white cells. So granulocyte, colony stimulating factor, macrophage, colony stimulating factor, granulocyte, macrophage, colony stimulating factor. So we get to see all those play out. Stimulates the growth of the hematopoietic stem cell in the bone marrow in response to inflammatory cytokines such as IL-1. Colony stimulating factors. So here's, right? Who wants to break this one down? Look familiar. Hopefully it does. Down here we got our little diff. Right here we got our little erythrocyte, our basophil, eosinophil, neutrophil, and our monocyte. So we don't have the lymphoid line, right? Because it kind of stopped up here with the lymphoid. This is the myeloid line. So we get erythropoietin, EPO. We get our granulocyte, monocyte, colony stimulating factor. This is our granulocyte proliferative cell, maybe. One of those, a blast in there. Whichever one you want to call it. Y'all know them, right? Y'all gotten that far? Am I really confusing you? I'm confusing you, or you gotten this far? Or are y'all just still on red cell proliferation? Where are y'all on hematology this time?
She hasn't gone through that yet. She
Oh,
said
no.
it confuses to go through this part.
Oh no. So y'all, we're, all right. Okay. All right, so we won't spend a lot of time on that, but we will talk about EPO, erythropoietin, okay, produced mainly from the kidneys. It regulates red blood cell production in the bone marrow. Maybe this will help you on your heme test tomorrow, maybe. Maybe you hadn't seen this, I don't know. Red blood cell proliferation is induced by erythropoietin. It improves our oxygenation in our tissue. So we see this with sometimes patients that are losing their red cells. Maybe they're under chemo treatment. They might get erythropoietin shots, and we have to do a red blood cell count before we do that shot. Do that a lot as outpatient. So to finish up, I know we're running a few minutes. We got just a couple more things to look at. I think it's a couple more slides, and then we'll be finished. I want to finish because we're going to do complement on Monday. We can do it. I think y'all can hang in there with me on complement. Love complement. So y'all got a big lecture to look forward to on Monday. Various therapy approaches when we're talking about cytokine and anti-cytokine therapies. So we get an infliximab. Remember, that's a monoclonal antibody. Anytime you see that ab there on the end, and what they like to do, what drug companies like to do is once they develop these, they like to give it a cute little name so they can put it on TV, and it'll be real catchy. So they'll change these names from where they started sometimes. But what we're doing is we're giving patients treatment to slow the cytokine storms down, to slow down the autoimmunities. So we have monoclonal antibodies that actually will be antagonists to the cytokine action, right? Go after it. So that's what we're talking about here. We're not gonna, infliximab, if I threw one of those on there, it wouldn't be what does it actually do, it would be in the approaches that a treatment for. Okay, so you want to see, IL-17 being blocked, right? We talked about that just a few slides ago as being that one that's seriously an autoimmune causer there toward the end of that little list we were getting excited about, because that was our T helper 17 doing that. Clinical assays, we're going to get into the ELISA technique. All right, it's going to be fun. We take a fun approach to ELISA testing. You'll set it up. We'll search for one or two things. We'll probably do 2 days. So we were going to start it next week. You look at your lab schedule, but since we got a test, we're not having lab. I really didn't want to, it just needs 2 days. So I want to do it on a Monday, Wednesday. We may prep for it next week, kind of get the stuff together. But what we do is it's a really cool little exercise where we actually take one or the two approaches. We can search for an antigen one day and then come back on Wednesday and search for an antibody. And we will set that into motion with an ELISA test, which is enzyme linked immuno assays. Okay, so you'll see that. So we will get into the ELISA technique of how that works. All right, and that concludes our cytokine lecture. You have your summary, which is very important. So you can definitely use that to study through the quiz when it's time, or you can study with that in the end chapter. So keep your PowerPoints for cytokines. I went ahead and printed them today. I didn't know if we'd get there or not. We didn't, which is fine. We got Monday, and then these 3 chapters will be on the test Wednesday by the end of the day. So I'll give you all day. And just know that I know that immunology is going to go better the second go because I saw how micro went on the first go. So I think you got the idea of where you need to focus your attention. And I think immunology should go skyrocketing back up into the high 80, 90 average on the next exam. And that's where we want to be. So y'all have a great weekend and see you Monday for compliments.